Hello everyone, my name is Urs. I'm an assistant professor at the University of Copenhagen, and I briefly want to introduce our study around fibroblasts in human gut-associated lymphotissues. Human gut-associated lymphotissues are these specialized immune priming sites within the intestinal wall, and they include, for example, isolated lymphoid follicles or Peyer's patches. And we studied these tissues based on surgical resection material coming from patients with colorectal cancer, where we took the healthy tissues, or inflamed tissues from patients with Crohn's disease. And then we went on and used our intestinal layer separation protocol, allowing us to obtain cleaned up lymphoid tissues, submucosa and mucosa, and then performed single-cell RNA sequencing, flow cytometry, and confocal laser microscopy. And if we check our single-cell RNA sequencing data, we found that there's broadly 4 different types of fibroblasts in the intestine. But the different subsets are present in very different ratios across these sites. And of course, this red cluster caught our attention because it's very much enriched in gut-associated lymphoid tissue. And digging a bit deeper, we found CD24 as a good marker for this cluster, and that was also confirmed in validation experiments, for example, using confocal laser microscopy, where we found protoplanin-positive fibroblasts in lymphoid tissues, indeed expressing CD24, but not in the surrounding lamina propria. We think that there's also further diversity within this cluster, and that's true for Peyer's patches, mucosal isolated lymphoid follicles, and submucosal isolated lymphoid follicles. And very briefly, we think that there are CCL19 and CCL21 positive T cell zone reticular cells at the follicle margins. There are CCL13 positive cells, which include complement receptor expressing follicular dendritic cells, but also other CXCL13 positive fibroblasts. There's a cluster which expresses CXCL14 at the subepithelial dome. There's in some of the follicles, we found a CXCL10 positive interferon gene signature cluster. And then in mucosal isolated lymphoid follicles in the Peyer's patches, we found rank ligands, so TNF-SF11 positive cells in the subepithelial dome. So we think that there's actually quite a diversity of fibroblasts, and all of them seem suited to recruit different types of lymphocytes and have the potential to activate them, for example. And lastly, we also wanted to check whether Crohn's disease has an impact on this landscape, or whether fibroblasts could possibly contribute to immune cell priming during Crohn's. And if we pick, for example, the T cell zone reticular cells and check differentially expressed genes, we see that there are some genes, especially around extracellular matrix production and leukocyte migration, for example, which are differentially expressed in inflamed samples. And if we then pick a couple of the top differentially expressed genes and make a signature out of this and apply it to all the different subsets, we can see that these changes are actually reflected in the other clusters as well. So overall, we think that these gut-associated lymphoid tissues have a very specialized and spatially restricted landscape. We also have CD24 as a marker for gut-associated lymphoid tissue fibroblasts. And we found that in Crohn's disease, we have largely the same fibroblast subsets, but these acquire an inflammation-associated phenotype. And that's it from me. I hope I got you interested in our study, and I also want to thank everyone who was participating in the study, the funding sources, and of course also the patients. Thank you.